Okay, so thanks. So today I'm happy to host a friend, Costas Lyssiotis. Costas is a fantastic scientist. He's an assistant professor at the University of Michigan. He holds a joint position in the Department of Molecular and Integrative Physiology and Internal Medicine and Division of Gastroenterology. So Costas did his PhD at the Scripps Research Institute in San Diego in the lab of Peter Schultz, where he developed his interest in chemical biology. Then he moved on to do his postdoc at Harvard Medical School in Boston in the lab of Lou Cantley, where he focused on the role of metabolism in cancer with a focus on pancreatic cancer, and where he discovered very nicely that Kras can reprogram pancreatic cancer cell metabolism by regulating key enzyme here, drawn here, GTH or GLUT1. Of course, Costas earned a lot of awards. I'm just listing few here. He got the Young Investigator Award from the American Gastroenterological Association. He got the Cancer Research Scholar Award from the American Cancer Society, the V Foundation Scholar Award, as well as the Aspire Award from the Mark Foundation. So I'm just listing a few, but this is an amazing publication, collection of awards actually, and of course publication records. So when I think about Costas, three things come to my mind. Besides his fabulous mustache, Costas, he thinks a lot about an enzyme called GOT, which has nothing to do with Game of Thrones. This is much more cooler than Game of Thrones. It's the glutamic oxaloacetic transaminase. His lab has done very nice work on this enzyme in the context of cancer using state-of-the-art metabolomics and isotope tracing. Costas' lab also thinks about the tumor microenvironment. They try to understand how stroma cells in a tumor microenvironment can support cancer cell metabolism and tumor growth. And there is a very nice publication in Cell Metabolism demonstrating that pyrimidines secreted by tumor-associated macrophages can actually participate to resistance to chemotherapy in pancreatic cancer. His lab thinks deeply about pancreatic cancer, and his research program seeks to translate basic understanding of cancer metabolism and how it is deregulated into new and targeted cancer therapy, with of course an emphasis on pancreatic cancer. And today he's going to talk to us about metabolic networks in the tumor microenvironment. I'm looking forward to his talk with a lot of isotope tracing and metabolic flux analysis. So welcome to us, and I'm going to stop sharing my screen.
Well, thanks, Isam, for that extremely kind introduction and for putting the slides together and giving some background. I'm not certain GOT is more interesting than Game of Thrones, so I will talk a little bit about GOT and then we'll let the audience decide later. So appreciate the invitation, Isam, you, Nav, Leon, Ali. And I'm looking forward to the opportunity. Looks like several talk interactions are lined up after the seminar. So I'm looking forward to getting to know colleagues at Northwestern better and appreciate it. All right, today's story is going to be, I'm talking about two unpublished works. All right, both of these are on bioRxiv if you want to follow along and get more information afterward. And in fact, I've tailored this talk for the groups studying mitochondria, the Shandell group and others at Northwestern in part, so that would be interesting. And then in addition, these are unpublished work, so love feedback and input on the science and the concepts and so forth. Like I said, if you want more information, I do a real good job of referencing. I think that's important in the bottom corner. And if you want to hear more about the lab, you can check us out either the lab website or the lab Twitter account. Let's see here. Okay. So one of the main goals of today's seminar is to communicate to you all the new concepts and the new work that's going on in the lab. And I know that this is extremely challenging in a virtual setting. And so I would ask, in order to make this useful to you all and to me, that if something's not coming across clear, that you interject. please speak up, turn your microphone on, ask a question, or if you're the really polite type, type it into the chat box, and Isa might ask that you monitor the chats, and if find a good time to interject. The talk I put together here is modular, and so if we get a lot of questions, I'd prefer that. Interaction is always a better way to learn, and we can I can easily drop sections of the talk. That's not a big deal. All right, so like I said, today I'll be talking about a recent work on looking at metabolic networks in the tumor microenvironment. I hope all the cancer biologists on the call appreciate that tumors are solid tumors consist of a number of different cell types that interact to create unique physiology. A common feature across all solid tumors is a disrupted metabolic profile. And so that's manifest by leaky and insufficient vasculature often, and it leads to gradients in access of cells in the tumor to nutrients, oxygen, the accumulation of metabolic wastes, pH gradients, and so forth. So how do the cells in a tumor deal with this? In my group, we think of the sort of the way that they're dealt with in three flavors, right? And three metabolic types of interactions. And I'll walk through those here briefly. Those which are cooperative in nature, those which are competitive in nature, and then a number of different interesting, and I'll talk quite a bit about this today, mechanisms of therapeutic resistance that emerge through metabolic communication. All right, so the first type is cooperative interactions, and I will describe more of these, but in essence, one could imagine a waste from one cell type, for example, from a cancer cell, could be considered a fuel by another cell type, and they could interact together so as to maintain, remove waste and maintain an appropriate nutrient profile that supports the growth and the bioenergetics of the tumor. So that's cooperative. The competitive types of metabolic interactions exist, or as we think exist, primarily to keep the anti-tumor immune system at bay. And so these are interactions between, let's say, malignant cells and the anti-tumor T cells, where the cancer cells will eat the nutrients such that, or catabolize the nutrients such that they're not available for the anti-tumor T cells, which similarly need nutrient fuels in order to proliferate and for effector functions. All right, and an area that we've jumped into a bit more recently is this concept of therapeutic resistance. And like I said, the two talks I'll go through today concern this, where metabolites can act directly through a variety of mechanisms to impair the activity of chemo and immune-based therapies. So if in addition to these sorts of interactions, I'd also like to put forth this concept that, and if you'll humor me, that metabolic communication, these intracellular metabolic communications inside of a tumor are every bit as important and maybe as prevalent as the sort of growth factor receptor interactions, and that we've really just scratched the surface on characterizing both the number and the utility of these in cancer biology. All right, so my lab works on these sort of interactions in pancreatic cancer predominantly. This is a representative histological image of a pancreatic tumor. And you don't have to be a histopathologist to appreciate that the cancer cells are a minority. So I've outlined one of the neoplastic areas here in this dotted line. And you can see that the majority of this tumor is actually stromal in nature. So most of that is activated fibroblasts and tumor-associated macrophages. Okay, and so we're curious how these sort of cell types are interacting with the cancer cells. And major questions based on this that come up in the lab, and these are themes that I'm going to talk about throughout, so I'd like to just plant these in your mind now, are could cooperative metabolic pathways, that is, like on the last slide I told you about where there's crosstalk between cell types, provide a source of nutrients or promote therapeutic resistance for pancreatic cancer? I didn't bring this up. Pancreatic cancer has the lowest survival rate of all major cancer types. It's around 10%. And so a big question in the field for a long time has been, why do these why are these tumors so resistant to therapies? And we think that part of that is related to their metabolism. Pancreatic tumors are also notoriously nutrient austere, and this is because there's very low vascularization of these tumors, and that is in part based on the physiological properties of the tumor. I won't get into that, but just knowing that they're nutrient austere, is this one of the reasons why pancreatic tumors are considered to be immune cold tumors? And is this one of the mechanisms by which the T cells are unable to attack the tumor? And as we consider both of these bigger questions, always in the back of our mind are how can we develop new drug targets and therapies based on these concepts? All right, so my lab's been working on this in this area now for about 5.5 years since I started at Michigan, and I think we've made some considerable headway. Like I told you earlier, I think we're just really the tip of the iceberg on characterizing the sort of metabolic interactions that exist within inside a tumor. And I'm going to go through briefly a little scheme here, summarizing some of those, and then also putting into context the stories I'll be telling about today. So my lab got interested in this in our first publication together with Alec Kimmelman's lab, where we demonstrated that pancreatic cancer cells are able to induce autophagy in cancer-associated fibroblasts, that when autophagy is induced in cancer-associated fibroblasts, they release copious amino acids, including alanine, and alanine is captured and utilized by the cancer cells to support both bioenergetics and biosynthesis. But what was really striking about this study, at least in my mind, is that we were able to demonstrate that blocking autophagy specifically in the cancer-associated fibroblasts, okay, so targeting metabolism in the stroma exclusively, was able to blunt the growth of pancreatic tumors, which I think really illustrates the metabolic support role of the stroma. All right, thereafter, we were working and continue very active collaboration with Wei-Ping Zhou's lab, and we found that regulatory T cells will infiltrate tumors where a variety of conditions will lead to their apoptosis. All right, so that happens with a number of cells, but what makes Tregs particularly interesting is that in death, they release these two ectonucleotidases called CD39 and CD73, which convert the ATP lost from the apoptotic cell into this potent immunosuppressive molecule, adenosine, and this molecule blocks the activity of cytotoxic T cells. We've also been investigating a number of different amino acid axes in pancreatic tumors. Recently published a paper on cysteine and methionine. You can check out the references here. And we have a very cool study ongoing now with Marina Pasca-Dimigliano's lab looking at the role of arginine in pancreatic tumors and how its catabolism by TAMs influences pancreatic biology. Isam mentioned this one. This is a study we published, I think, most recently here. It's a bit of a different take. Again, this is the therapeutic resistance angle. So postdoc in the lab, Chris Halbrook, he's now running his own group at the University of California, Irvine, demonstrated that pancreatic cancer cells polarize macrophages into this tumor-associated macrophage subtype, reprograms their metabolism, causes them to release a number of different pyrimidine nucleosides, including, and the one we focused on for the sake of the study I'm going to tell you about here is deoxycytidine. And that's because deoxycytidine, hopefully you don't have to be a chemist to appreciate, looks a whole heck of a lot like gemcitabine. And we demonstrated that through molecular competition, deoxycytidine from TAMs is inhibiting the activity of gem. We have another paper that's now, we just bioarchived this one, co-submission with Katie Welland's group. This is a cool new concept where we demonstrated that CAFs can feed cancer cells extracellular matrix, and in particular, the one we focused on here was the glycosaminoglycan high hyaluronic acid. And so pancreatic tumors are chock full of high hyaluronic acid, and this is actually a carbohydrate polymer consisting of repeating units of glycanic and glucuronic acid. And so Peter found that that can be actually a nutrient fuel produced by CAFs and utilized by cancer cells. Like I said, this is with co-submitted with Katie Welland, and if you're interested in checking that out, there's links here, or you can check out the website. All right, so that's sort of the backdrop work that we've done in the past, or even some ongoing. And But what I'll tell you about is in today's seminar is two different unpublished stories. And like I said, these focus around mitochondrial metabolism. Ultimately, they do involve the GOTs, so Isam will be impressed. Unfortunately, Isam, we don't have any new studies on nucleotide metabolism to share, but I hope Nav appreciates, and the Shandell group appreciates the work we're doing on mitochondria here. So the first story, this is spearheaded by Sam Kirk. He's a graduate student in the group. And Sam found that pyruvate is released by pancreatic calves. There's also pyruvate in circulation, which we think is probably equally important, but this is where the story started. And this pyruvate is able to mediate resistance to mitochondrial targeted therapies in pancreatic cancer. And so I'll tell you a little bit about how that story evolved from a GOT perspective, actually. And then the second story I'll tell you about is more work from Chris Halbrook. And in this instance, what Chris demonstrated, we were looking at how potentially interclonal metabolic heterogeneity existed in pancreatic tumors. And then to look if these different malignant clones, their differing metabolic programs could cooperate in some fashion. Ultimately, this started as a basic, real basic concept, but I think it's evolved into something that's we feel is much more important as it relates to the role of asparagine. And the role of asparagine in particular, what we demonstrate is one cell type releases asparagine, and like pyruvate, this is also able to mediate resistance to mitochondrial targeted therapies like fenformin. So that's sort of a big picture observation, but also this is something that's therapeutically tractable, and we have really convincing evidence here in preclinical models because you can target asparagine in circulation. This is a therapy that's been around for decades. It's called asparaginase, and it brings down circulating asparagine levels, and that could be potentially combined with mitochondrial targeted therapies. All right, so this work, I will say, is we co-submitted this with Heather Christoph's lab. She came to similar conclusions. I'll tell you a little bit about Heather's work too as we go through this. All right, so the first story starts as I normally don't have such a nice introduction from a host or whomever, but Isam put this into context with the GOT. So the story starts with the malate-aspartate shuttle. Here's GOT1 and GOT2 right here in the center. The role of the malate-aspartate shuttle is to take reducing potential generated in glycolysis and move that into the mitochondria to fuel the electron transport chain. So NADH is unable to cross the mitochondrial membrane, and so through evolution we've evolved this really elegant mechanism by which that reducing potential is shuttled through metabolites into the mitochondria. So when I was a postdoc with Lou Cantley, we found, and this is working with Alec Kimmelman's group and Jay Sun, that the malate-aspartate shuttle is rewired in pancreatic cancer such that glutamine-derived carbon is brought in through a partial malate-aspartate shuttle, and then the reducing potential generated from glycolysis, it is similarly put onto malate, but that malate then is oxidized to pyruvate and produces NADPH concurrently. And the reason for that, I won't get into too much of the details, is because this alpha-ketobutyrate is not available for co-transport, and as it's an anaplerotic source here. So that's a little backstory. You can check out that paper if you want to hear more about it. But when I started my lab, we were interested in, okay, could any of these enzymes in this pathway serve potentially as a therapeutic target? And so we were pursuing that when I got the lab off the ground. And without really knowing which was the best one, we took an agnostic approach, and we said, all right, let's just go after all of them. So this was a pretty big endeavor, but I think it's proven fruitful. We have different programs now on each of these enzymes. I'm going to tell you about that related to GOT2 today. So this is where it started. We made doxycycline-inducible hairpins that could target each of these individual enzymes. This is one of the highly responsive PDA lines. You can see that basically the cells don't grow out in colony-forming assays when you knock down any of these enzymes, quantitated here, just to illustrate sort of conceptual structure for this research program. I'll focus in now on the GOTs, and these two graphs were generated by different people, so they're not exactly the same, but presented exactly the same. So I'll just briefly walk you through those. We have colony formation on the y-axis. So this is GOT1 here where my cursor is. If you use either of two independent hairpins, you can see real differential response to GOT1 across a panel of pancreatic cancer cell lines, but there are some that are really quite responsive. The same is true of GOT2. So here, rather than being normalized, the non-targeting hairpin is presented as its own bar, and same sort of concept applies, right? GOT1 and GOT2 both basically result in this spectrum of dependence across a panel of pancreatic cancer cell lines. Okay, so in moving this project forward, we said, all right, well, let's take these into in vivo models and see what we get. In the case of GOT1, this has been pretty consistent. We've published some of this work, and it's not knocking your socks off in the sense that we do have tumor stasis, much like we see in vitro, but we're never able to get tumors to turn the corner. So there's definitely something interesting happening here. This is not a holy grail drug target, but anyhow, what it did present is an interesting contrast, because when we did the same with GOT2, this is probably not the punchline you expected. What we saw is absolutely no phenotype. And so we said, all right, well, these are two pancreatic cancer cell lines. So maybe when we take so I should mention, what you have on the y-axis is tumor volume. In the black line, this is an isogenic set. There's no doxycycline in the reds. When you bring on doxycycline, and that knocks down the GOT enzyme in question. No growth retardation with the GOT2 knockdown. So we said first, all right, maybe this has to relate to some leakiness in our system. No, we're able to really keep the GOTs down quite well. So that's not what's attributing to this lack of growth. So we said, all right, let's test a bunch of other pancreatic cancer cell models. We've tested 7 independent models now, and when GOT2 knockdown has absolutely no impact on tumor growth in these immunocompromised settings. So these are all human cells, all immunocompromised settings. We can talk more about the immune system at the end. It would definitely be an aside. So I thought this was really interesting, and a naive graduate student joined my lab, Sam Kirk, and I talked him into taking on this project of differential phenotype between in vitro and in vivo biology. This was, 3 years or so ago and I still think there's a lot of excitement in understanding what is it that's different about the culture conditions we use in vitro versus what, a cancer cell or other cell types are seeing and experiencing in the tumor in vivo. So I got Sam excited about this project. Again, the punchline is GOT2 knockdown has no phenotype. And so Sam said, all right, maybe something being released from cancer-associated fibroblasts are impacting the growth of the tumors in vivo. And since those aren't present in vitro, that's where the action's at. I should also just show you these are xenograft tumors. I think it's important to demonstrate here that there are indeed CAFs in these tumors. Okay, they're pretty stromal. So I wouldn't be telling you about this line of logic if it hadn't held up. And this is some of Sam's data here. You can see that when you knock down, so this is again a colony forming assay, some representative colonies here. When you knock down GOT2 with 2 independent hairpins, the cells don't grow in normal DMEM, but if you put on CAF conditioned media, they grow through it. Effectively a full rescue. Excuse me, this has nothing to do with the modulation of GOT2 expression. You can see that in the presence of CAF conditioned media, there's still potent GOT2 knockdown. Oh, you know what? I'm going to skip this one in the interest of time. I'm already running a little long here. So we demonstrated that the conditioned media from the CAFs was dose dependent. And then we went one step further and did something that my lab's become quite keen on, and that is to boil the media or run it through low molecular pass cutoff filters to see if the activity is retained in the conditioned media. So in this very stripped down assay, again, we're using conditioned media that has been treated in some way, we're still able to retain the activity. That gave us some confidence that what we were looking at was a metabolite in nature. And so my lab has, since starting, put together through support from a number of different trainees in the group, what we call our SNAPSHOT Metabolomics platform. This is really the workhorse platform of the lab. It's extraordinarily robust for the metabolomics aficionados. Glad to tell you more about this. You can put thousands of samples through the same column, and we generate our samples from virtually anything you can get out of an organism: cells, tumors, organs, organoids, sera, feces, you name it. And from that, we very reproducibly are able to get what I call a snapshot of the metabolome at a given point in time, under given circumstances. And we get a sense for what's happening in the major metabolic pathways. So Sam took some the conditioned media from his calves, and he ran it through our snapshot platform. And what he saw come out was, so these are 3 independent preps. And you don't need to remember all of these, just so you can see that there's some interesting differences, generally speaking. At the top of the list was aspartate and N-acetyl-aspartate. So he said, all right, GOT2 makes aspartate. This is a no-brainer, slam dunk. We tried aspartate every way to Tuesday to get it to rescue GOT2 knockdown, and it did not. And I was convinced that was the answer. I thought this was going to be a real quick paper. So let's just show that aspartate, publish this thing, and be done with it. But aspartate is not the answer. We were unable to rescue the aspartate. So Sam went back through and looked for other metabolites. And at this point in time, some studies were coming out of several different labs: Van der Heyden lab, Sabatini lab, and they what they saw. Well, I'll give you a little more context on their work in a second. But what Sam saw, led by those studies, is that pyruvate was another metabolite that was abundantly released from the calves. Okay, so it's at the top of our list here for fold change relative to DMEM. We quantitated that, and this is maybe on the higher end of quantitation. We're really more in the 250 to 500 micromolar abundance range from the calves. So the calves are releasing, based on our conditions, 2 days of culturing, about 250 to 500 micromole of pyruvate. All right, and I tell you that number, it's important for 2 reasons. Sometimes people don't appreciate this. The first is we want to be able to normalize across our assays. So if we're using calf conditioned media, we want to use the same dose of pyruvate when we go in and try to do an even more stripped down assay to make sure we're working at the same concentration. And secondarily, we want to be mindful if we're working in a physiological range. And the amount of pyruvate in serum is around 50 micromolar. So we think we're well within range. Alex Muir, I don't know if Alex is on the line. I realize he's at a different Chicago University, but nevertheless, I don't know how close you guys are. So Alex showed that in pancreatic tumors in the interstitial fluid that the amount of pyruvate is on the order of hundreds of micromolar. So we're really right in the ballpark here, which further confidence that we might be barking up the right tree. So Sam took pyruvate directly. In this case, he used 1 millimolar. I'll show you later. We can use lower concentrations. Let's see here, 1230. And he's able to achieve a near full rescue, again, just with pyruvate instead of the calf conditioned media. So that brought us to, well, what is it that pyruvate's doing? And I'm going to distill a lot of experiments into, by telling you all the negative stuff just right off the bat here, and then going into what did work. So pyruvate can be converted into a number of, has a lot of fates and functions. It can be converted into a number of different metabolites or metabolic pathways. In doing so, it also results in the balance of other, let's say, fluxes in a cell. Okay, so pyruvate's converted into alanine. This can be one way by which nitrogen is balanced. Pyruvate can be converted into acetate, and in doing so, detoxify ROS, TCA cycle. It's probably maybe most classical is conversion. It's reduction into lactate, and then concurrent oxidation of NADH. So we tested all of these metabolites down here. None of them were able to rescue. We also tested an inhibitor of pyruvate transporter into the mitochondria that did not prevent rescue. We messed with the nitrogen and the ROS. None of that worked. So we ended up spending our time here on the NADH balance. And part of that was also guided, I will say, by some studies that are noted here in the references. Okay, so a lot of these concepts we were working on right when other people were publishing really great papers, and so they, standing on the shoulder of giants, let's say that, inspired by their great work. But as we were building toward our mechanism, so let me show you some of the data that got us there. The first thing Sam did was to take, these are the PDA cells, now cancer cells, knock down GOT2 directly in the cancer cells and submit the metabolites for metabolome profiling. You don't need to know all the enzymes that are presented here on this list, just suffice it to say, I drew the schematic for you. When you knock down GOT2, there appears to be, this is glycolysis here, an inflection point in glycolysis at this white line, all right? And that is the enzyme, this inflection point is at the enzyme GAP-DH, which reduces NAD to NADH. All right, so when we knock down GOT2, there's a decrease in all of these, and there's an increase in all of these. That reflects NADH stress or an accumulation of NADH such that the GAP-DH reaction cannot proceed downwards. All right, so that was the first piece of evidence. Sam also saw reflective in the metabolomics, a disruption in the NADH-NAD level. That's also shown here, right? NADH goes way up when you knock down GOT2. It's consistent with its role in the Malate-Aspartate shuttle, also point that out. And then to more specifically tie a role of NADH to this GOT2 phenomenon and the role of pyruvate in its rescue, we took two orthogonal approaches. The first was lifted from this Luke Sullivan paper in Matt Vanderhein's lab, where he showed using, like I said, an orthogonal metabolite, alpha-keto acid, alpha-ketobutyrate here, instead of pyruvate. So alpha-ketobutyrate does not enter metabolism in the same way that pyruvate does, but like pyruvate, it will oxidize NADH, okay? And in doing so, restore NAD levels. And AKB is not as great as getting into cells, and we can talk about some of the nuances, but the fact of the matter remains that we were able to achieve some degree of rescue with alpha-ketobutyrate. And so we thought that this is probably it. It's probably about NADH, NAD. So this is a totally orthogonal approach to pyruvate. Again, this only looks at the NADH role of pyruvate, not its other metabolic fates. Though I did tell you that we blocked mitochondrial pyruvate carrier activity, and that did not block the pyruvate rescuing activity. For the aficionados out there, if you didn't follow that one, just ignore it. It's a lot of information. Okay, the next thing we did is, again, it's an orthogonal assay. And this here, what we're doing is oxidizing NADH. This is a trick we lifted from Vamsi Mutha's group. They can express this lactobacillus NADH oxidase. And what it does is it takes the electron on NADH and puts it onto oxygen to make water. So basically it's regenerating the NAD without having, getting rid of this NADH stress, let's say. This is a cool system. You can express this in the cells in either the mitochondria or in the cytosol, and you can do so in a DOX-inducible manner. So Vamsi already made all of these and put them on Adgene, and we just grabbed them. So let's cut to the chase here. So this is the Western on the left is confirmation that the system's working properly. But I think you'll appreciate the fact that we're able to do a near complete rescue using this LB-NOX as evidence that it's working properly. Also of note, we can get into this more if question and answer session. This was unique. So LB-NOX was able to rescue GOT2 knockdown, but only the cytosolically expressed form, not the mitochondrially expressed form. And what, like I said, LB-NOX is removing that NADH stress. So what we concluded, let me see. Yeah, I have first summary slide here, that pyruvate is microenvironmental pyruvate, part of which is coming from CAFs, we presume, is able to preclude the need for GOT2 by oxidizing NADH to restore NAD pools. And this is happening in the tumor microenvironment, and that's why these cells are not dependent on GOT2 for their growth. All right, I'm going to try to jump slowly here. And I don't have people like directly bugging me. I just talk slow, perhaps. Let's say that. All right, so why is this relevant or therapeutically important? There are a number of mitochondrial targeted therapies that are moving forward in oncology, in clinical trials. So fenformin has entered some clinical trials. This is the sister of metformin. I'm sure many people know. If you put fenformin on pancreatic cancer cells, it impairs the growth, and that can be restored by either CAF conditioned media, so that's in the green line, or in directly with pyruvate. So that the blue line was known, but we demonstrated this can happen by CAFs. And what we put forth is that this is a major mechanism of therapeutic resistance that's going to exist a priori against targeting the mitochondria in CAF-rich tumors. We also demonstrated that fenformin was one complex 1 inhibitor we tested. We also used this other complex 1 inhibitor from the MD Anderson group, and we see similar phenotype. So this is a very busy slide. Let me just try to hit the punchline here. We then wanted to know, all right, could we sensitize pancreatic tumors to mitochondrial targeted therapy by blocking their ability to access pyruvate? And we did that by going after the pyruvate transporters. So there's a class of these. They're called the MCTs. There's a ton of promiscuity amongst these, and there's definitely redundancy in their ability to transport keto acids. MCT1 we found it's thought to be the pyruvate transporter, has a lower Km for pyruvate. We didn't see expression of MCT2 or MCT3 on these pancreatic cancer cells. So we went after MCT1 and we used a CRISPR approach to knock it out. And what you can see here is that, so right here, this is just in normal DMEM, these three bars. If you add pyruvate, you're able to, into these GOT2 knockdown cells, this gray bar that I'm pointing at, you're able to rescue growth. If you knock out MCT1, you reduce the ability to rescue the growth, right? Which means pyruvate's not getting in as well. We think that at 1 millimolar, there may be some redundancy from other transporters. So once you get closer to physiological ranges of pyruvate, like I said, this is in the hundreds of micromolar, you can see that that effect is even more profound, right? The MCT1 knockout to prevent the growth of these cells. And then you see activity at 50 micromolar pyruvate as well. So that's where the story's at now. I'll tell you, we're trying to take those concepts. They work great in this in vitro system. It's been trickier getting it to work in vivo, but we are trying a couple different approaches. And the punchline from this is that microenvironmental pyruvate, like I said, circulation and from CAFs, is able to support the growth when under NADH stress in a cancer cell. And so that can be imposed by GOT2 knockdown or inhibition of complex 1 we demonstrated. All right, I will, if there's a pressing question, like I said, please do stop me. Isam, I don't know if there's anything coming in the chat box. I don't have good access to that. We're good? Okay.
No questions so far.
Okay, I'll keep trucking here. And I'll try to, see, I'll stop in 10 minutes so that there's...
I have a question.
Yeah, okay.
Did you look at the serine glycine pathway? Because PHDH uses NADH, and you have increase in the NADH-NAD ratio. What happens to the serine glycine pathway in your GOT2? Okay. Yeah, it's a great question. So in our experience, Isam, I'll tell you, in the human pancreatic cancer cell lines, they don't do any serine biosynthetic pathway activity. They get all of their serine from, now granted, there's going to be a lot of caveats here, okay, but all of our cultured cell studies where we use medias that have serine and glycine in them, they take all of their serine and glycine from the media. The amount that they derive from glucose is on the order of 1 or 2%. So we don't think that that's a big push in these pancreatic cancer cells. The reasons for that aren't clear. It's never been something I've been terribly interested in. And so for that reason, we didn't go down that, that path. It may be different in vivo, but nevertheless, we didn't go down that path. Okay, so the next question we started asking, it's a simple one, is there's
genomic complexity in pancreatic tumors, in all cancers, that results in different clonal populations? And could these different clones, which in pancreatic cancer is well documented, you have your epithelial type and you have your mesenchymal type, could they be interacting at the level of metabolism? So that's a question Chris set out to ask when we got this story off the ground. And we worked with our friend Klaus Jorgensen at CRUK Manchester. He generated clonal cell lines from a murine pancreatic tumor that we were able to profile metabolically to look at differences among the clones, both at the intracellular level and at the extracellular level, to see if there was communication. Like I said, I'm moving through this pretty quick. This work was, Chris was supported quite considerably by a really talented technician, Galloway Thurston. Okay, so we submitted those samples for our profiling. In looking at those clonal populations, really, they broke into two groups, both at the intracellular and the extracellular level. I'm not going to belabor this because I want to get into the data a little bit further. So here's the extracellular metabolome profiling. You can see that there are differences in the amount of amino acids and nucleic acids released. Those were really two major categories of molecules. All right, and so if we take this, like I said, somewhat naive idea, could there be two subtypes of cancer that are supporting each other at the level of metabolism within a metabolically stressed pancreatic tumor? To test this model, we said, all right, let's apply some inhibitors of glycolysis and the mitochondria to each of these cell types to see if, independently maybe they're hurting, but together they're working better. And we saw that, we're going back to mitochondria here again, like I said, targeting this talk to you mitochondria folks. What you can see here, 2-deGC glycolysis inhibitor, there's really no difference in sensitivity between the two groups. But if you look at a variety of different, let's say, mitochondrial targeted therapies, as a pan-transaminase inhibitor, complex 5, complex 1, you see that group 2 is more sensitive than group 1. So then we got into the question, I stole my punchline a little bit here, but then we jumped into, could group 1 support group 2? So group 2, sensitive to inhibition of the mitochondria. Group 1 was resistant. Well, so what happens if you grow them together? And we did this, we used these fluorescent labels. The reason we jumped into fluorescent labels initially was because we were using metabolically encoded sensors and the IncuCyte platform, but it turned out that we could get the answers we needed just by using a fluorescent label. And so that sort of work ended up getting pushed to the back. But what hopefully you can appreciate here, let me get to the punchline. If you grow these two cells, so a unlabeled group 1 with a labeled group 2, or a labeled group 2 with an unlabeled group 2, we grow them at roughly 1-to-1 ratio. At endpoint, they maintain a roughly 1-to-1 ratio. And then if you treat with ligamycin, you can see that the group 2 cells will die when cultured with other group 2 cells. But when cultured with group 1, they survive. This is with ligamycin. We extended that also to fenformin. All right, like I said, I'm moving pretty quickly here. Happy to go through the details in a little more depth when we talk about questions, but I want to leave some time for that. So I'm distilling a lot of data. We looked at co-culture directly. Was that required? Was this a secreted factor? So co-culture was not required. It was indeed a secreted factor. And then we said, all right, like with the previous study, could you boil away that activity? And when we boiled the conditioned media, the activity remained, giving us some confidence that we were looking at a metabolite. So we went back to our earlier profiling. We said there was a lot of action with amino acids. Could this just be an amino acid? And could it just be aspartate or pyruvate? And so what we ended up finding is that it was an amino acid. It wasn't aspartate. We were surprised to see that. We were actually able to recreate the activity of group 1 protecting group 2 from mitochondrial inhibition by feeding the cells with asparagine. This was, I still don't know if I fully wrapped my mind around this. And I'll tell you why here. Oh, before that, let me just give you a little more context. So that was true here where we're able to rescue complex 1 inhibition with exogenous asparagine. We demonstrated that this wasn't true of the only true of the murine cell system. We think we've stumbled on something that's bigger, that is more of a generality. And we illustrate that here using a mouse and human pancreatic cancer cell line, another human pancreatic cancer cell line, and 293Ts. When Chris was presenting this work at a meeting, maybe even a couple of years ago now, Heather Christoph's group had very similar data, and she was working in a completely orthogonal system. Her team illustrated that they were able to do this with rotenone, different complex 1 inhibitor, and here N is asparagine, if you haven't memorized your amino acid codes. And like we see, where asparagine is able to rescue fenformin, they're able to rescue rotenone with asparagine. So we think that this is really some sort of general phenomenon. And so what does that mean? This is where been having trouble wrapping my head around this completely, but I think we have some pretty convincing data now that's pushing us in the right direction. And let's see, I'm gonna talk about the model here, and I'm not gonna walk through the data, but just really give you the big picture. So this is a model put forth by David Sabatini's lab and Matt Vanderheiden's lab, where they ultimately ascribed a function to aspartate as being rate limiting for proliferation, and its production serves both the purpose of supporting biosynthesis and being produced through respiration, thereby removing electrons from the system. So what we're able to demonstrate is, and they can rescue complex 1 inhibition with aspartate. I'll say that. And the role is aspartate is used downstream to support biosynthesis. So what we found is when you inhibit respiration, we can rescue with asparagine. So how does our data fit into this? And an important piece of information, if you don't know, asparagine is not converted into aspartate in human or mouse cells. All right, the systems we're working with. If you give them asparagine, and we demonstrated this with C13 tracing, okay, it was already known, but the reviewers asked us to do it. We're not seeing any asparagine being converted into aspartate. So how then is asparagine able to rescue complex inhibition? How does that fit into this elegant model proposed? So what we think is that the asparagine, there's a big pull on cellular aspartate to make asparagine. If you give the cells asparagine, you're relieving that strain to make aspartate. And so the asparagine can be used itself to make as an amino acid to make proteins to support proliferation. But there's also a big pull on asparagine because it is used as in antiporters. So it's pulled out of the cell to bring essential amino acids into the cell, like arginine. And in doing so, it is responsible for activating mTOR. So we think that this sort of dual role of asparagine, both by reducing the strain put on the production of aspartate, is able to support cellular biosynthesis through this mechanism, while allowing the remaining aspartate to be available to do the things it likes to do, like produce proteins, but also produce pyrimidines. Okay, so that's the model. I do have some data to support that. If you guys want to check it out, it's here on the bioRxiv. I won't walk through that all here. I want to finish by mentioning this last point, because I think, starting with this relatively simple model, we were able to find discover some important biology, but that also has therapeutic utility because of this therapeutic enzyme that has been in oncology now for decades, called asparaginase, which depletes extracellular, or rather depletes circulating asparagine. Here we're using 2 different pancreatic, immunocompetent pancreatic tumor models. In the first, you can see that either asparaginase or fenformin as single agent has modest activity. The combination really blunts tumor growth. This is a sub-Q tumor. This is an orthotopic tumor. We're able to shrink established tumors. We've now extended this to 4 or 5 other models, and it continues to be consistent. So I think there's really some utility here. There's even an ongoing clinical trial now looking at asparaginase plus gemcitabine in pancreatic cancer, really set the stage for us to consider this as a new therapeutic modality for pancreatic cancer. Like I said earlier, there's not a lot of options for that disease. Okay. I have some take-home messages here relating to hopefully that you appreciate the points I made early on in this talk, that metabolic communication inside of tumors is that we've really just reached the tip of the iceberg in discovering all of the ways that metabolism can support tumor growth through intercellular exchange, and that these may serve as important, or in some instances more important, roles than the growth factor receptor communications pathways that we're more familiar with, that this exists in that this can exist in cooperative fashions, that this can support the therapeutic resistance of a tumor, and that there is also roles in immunotherapy. So that's the talk. Let me take one more minute to thank my team here. So this is us pre-COVID. This was, these were the good old days when we go outside and get together. First, I'll point out Sam Kirk here. He's actually a floating head. I don't know if you noticed my Photoshop skills. Sam was at his honeymoon during this picnic. But so Sam led the first study on GOT2. The second study was led by Chris. Chris got a ton of support from Galloway. Chris is now running his own lab at Irvine, and Galloway went to med school at Michigan State. We have an awesome team of collaborators. None of this would have happened without them. I can't thank them enough. It's a long list, so I won't go through that. Thank funding sources. Thank everyone here for their attention, and Isam for the invitation, and I'm delighted to take questions. And I also look forward to interacting with people more during the meetings hereafter. Thanks.
Thanks, Costas. I think we have a question. Mark Mendilo.
Hi, Costas. That was really amazing. Fantastic talk.
Thanks, Mark.
I had, so the last half, I get the sort of biology that you stumbled upon, but I was a little curious about, you initially, I think maybe I kind of missed something, found this in a model of intratumoral heterogeneity. And I guess, is that correct? So you had one cancer cell type within, I guess these were mouse murine cancer lines that provided, the asparagine. What was that? Did I get that right? And what was that heterogeneity? Was that an epigenetic? Is this a plastic metabolic state, or is there? Are these? Did I get that right?
You're 100% right. Yeah, I don't even know. I don't know if I want to reiterate, because you hit all the main points. This is from a mouse model where the genetics have been set by RAS and P53. That from that, if you derive clonal cell lines from a parental tumor line, that they fall into two different metabolic subtypes. So Klaus's group they found within that 3 transcriptomic subtypes, and our metabolic subtypes sort of encompass one and then a pair. And yeah, we think we don't have a good characterization of it yet, but we think that it relates to epigenetic plasticity, and we see similar in human pancreatic tumors where there's transcriptomic subtypes have been described. It's not a perfect match, but we think that our group 1 subtype represents the more bioenergetic quasi-mesenchymal subtype, and that our group 2 subtype represents the more classical subtype. And there is documented quite a few studies on how those are epigenetically maintained and can interconvert.
But yeah, and in your work, it looked somewhat stable because you could isolate these cells for a while, I guess, and they'd preserve, preserve their unique activity. But it is a plastic thing where over time the original clonal heterogeneity is recapitulated. So
we have not demonstrated that, I will say. Maybe I did a little bit of hand waving there. We think that it is epigenetic plasticity, that it gets set. And then once it's set, there's probably some sort of cues that can bring them back or interchange them. We don't know what those are. We haven't found them. To be honest, we haven't looked for them.
Right.
But based on the fact that they have the same genetic makeup, I think that's probably the main driving factor for coming to that conclusion.
Okay, thanks. Great.
Okay, moving to the next question. Paul Schumacher, have a question?
Yes, first, Costas, thank you so much for just an incredibly inspiring, interesting seminar. And it's especially welcome in these mind-numbing times of working from home and being cut off from daily contacts with colleagues. So thanks again. I think we're going to have a chance to talk later, and I'm looking forward to that.
Oh, good. Me too.
So I have to ask you an annoying question. So I apologize for this, but you reminded me of how important mitochondrial interactions are with events in the cytosol in the context of cancer. And you and many others, including myself, often use antibiotics like doxycycline to turn on short-in constructs. And doxycycline, and indeed, the antibiotics that we use in cell culture medium are all mitochondrial inhibitors. They inhibit mitochondrial protein translation and other functions. So does it ever worry you that there's a possibility that some of the effects that we're seeing are in these sort of experimental conditions are influenced by the partial mitochondrial inhibition that you get when you're including these antibiotics in your cell system?
Yeah, that is a great question. So we've gone to lengths to convince ourselves that it is not involved, and because that is such an important caveat of using these systems. So I'll say first, we've done, this is 1 microgram per mil is our doxycycline dose that we use in cell culture. And in cell culture, we don't think that's relevant. We've done metabolomic profiling. We always include a dox control. We've done seahorse profiling. And I think it's just a low enough concentration that it's not impacting the metabolism. Others have reported that it can at that concentration, but in our pancreatic systems, it seems not to be having an effect. Now, in our mouse models, the way that we deliver doxycycline is in the chow. And we find that that is the most reproducible way to give the animals the doxycycline. So we haven't gone to the lengths of determining what the concentration is there. But the way we've convinced ourselves that dox is not related to the phenotype, well, first off, the GOT2 knockdown in vivo doesn't have any effect. So we're not getting any action from the dox, but we have also we've used CRISPR with GOT2 since we did that later, because that was at the point in time when we learned that you could grow GOT2 knockout cells with pyruvate. We didn't know that starting in those studies. So when at the end we used pyruvate to grow GOT2 CRISPRed cells. But with the GOT1 system, it still remains a stumbling block at times. But I think I've done everything I can to convince myself that if it is playing a small role, it's not driving the phenotype.
Okay, thanks very much.
Yeah, we can talk more about it. It's definitely something that keeps me up at night.
Okay, last question. I think time is up. Nav?
Costas, just a real quick question. You didn't touch it. You alluded to this a little bit in your talk about an intact immune system. So as you know, at least some of the studies you showed were with the GOT2 knockout versus GOT1 were in human cells. The reason I bring the immune system up is, as you know, our friend Alec Kimmelman and Kivanch both published back-to-back papers in Cell Metabolism. I think they're online now, where they show that if you look at in vivo dependencies of what genes are essential, right? In vivo, neither GOT1 or GOT2 scored for either one of them. Now, the caveat there is it's one cell type they use, right? The immune AP cells. It's not, so I just wonder, what do you think is going on there?
Yeah, it's a great question. I won't touch GOT1. I'm not sure what's going on with GOT1. But with GOT2, and we're working with Mara Sherman's lab, and I feel that I have the right to share these results because she's also presented them publicly. And like I said, we're collaborating. We made the GOT2 floxed mouse, and we've been pursuing that. But if you knock out GOT2, and we found this out after we did the human studies, but if you knock out GOT2 in murine cells that are syngeneic, such that you can put them back into the pancreas, those cells will not grow a tumor. So maybe kind of squashes some of the novelty of our human work. Hopefully the metabolism stands on its own, but in the murine system with an intact immune system, if you put GOT2 CRISPRed cells, which we grow in the presence of pyruvate, back into an animal, they're rejected by the immune system. And we're working now on determining what that is, what the mechanistic specificity of that is. And I don't think Mara has shared that publicly, so I won't go into that, but that's, why isn't Kimmelman and Kavanch seeing that?
Yeah, my own.
Curious, I don't know.
I'm glad you're doing the GOT2 floxed. I mean, run that through a proper mouse model, right? Which you will.
Yeah, we have the GOT1. Listen, I'll also put this out there. I know that you love working with these kind of enzymes and genetic mouse models. We have the GOT1 and the GOT2 floxed model. We have those in the KPC that's cooking. KPC, KRAS, P53. C is the pancreas-specific Cree. If you ever want to fool around with those in any of your systems, they're on the table.
Good. Those will be very important experiment. Great talk.
Thanks, Nav. Appreciate it.
Everybody.
Okay, I'm sorry about the other questions. We are running out of time. So thank you, Costas, for this amazing talk, and looking forward to talk later.
Me too. Thanks so much. So what's up now, Isam? Do I have